So if we could ask Dr. Voronova and Dr. Phillips to come back in, and we'll have a roundtable here. And I actually have a couple of questions in the stack that are addressed to both of you. So Dr. Voronova, this is a general question. What is the role of resident neural stem cells in adults if they are inefficient in regenerating the brain?
Right. So I didn't have enough time to go into all of these details, so thank you very much for asking that question. Our young brain is much better at regeneration, obviously, than an adult brain or an aging brain. So that's one component to consider. And of course, neurodegenerative disorders afflict more of an adult population than aging population. So that's one thing to consider. And it's highly debatable currently in the field, but if I quote some of the studies that use 14C atomic bomb, studies where they've actually taken a look at postmortem brains, have tried to figure out if some of the neurons or brain cells were regenerated, then there's actually some evidence that we actually generate 800 new neurons every day in our human brain. And so whether that actually translates to some of these oligodendrocytes, I don't know at the moment, but it was shown also using a similar approach that in MS patients that have very, very progressive state of disease, and they succumb to the disease in just a few years, that there is very minimal regeneration of these oligodendrocytes. So there is potential in them. I think it's just a matter of really waking them up and shaking them up. So thank you very much for that question. It was a really important one.
A quick follow-up about multiple sclerosis. Is enough understood about the mechanisms of that condition that you can see that your work with oligodendrocytes will fit into that picture?
Can you repeat your question again? So with multiple sclerosis?
So, yeah, because you mentioned multiple sclerosis as a possible target for the work that you're doing. But is the understanding of the actual mechanisms of the loss of myelin in multiple sclerosis like adequate enough, well understood enough right now that you can sort of see a path from your work to helping with that?
That's a very loaded question, but a really great one. So, of course, you're hitting it right on the nail. There's actually a few studies that came out just last year or 2 years ago that now implicate that some of these progenitor cells that I talked about can have immune function. And that has sort of opened the can of worms, because we always thought that in MS, the immune system attacks the oligodendrocytes, it attacks the myelin, which leads to neurodegeneration. Now that we've discovered that these oligodendrocyte progenitors in both humans and mice with MS can actually have some immune properties, the question is now whether they start the degradation, which then leads to immune cells infiltrating in. So it's actually a really active question in the field right now. And my lab is one of the ones that is trying to sort of pitch in and produce some of the data proving or disproving that hypothesis. So I think there's going to be a lot in the future, but it highlights the complexity of biology and how we need to really attack it from an interdisciplinary perspective. It's not enough just to know about immunology or just about neural stem cells. I think we have to have a sort of a continuum of understanding here.
Well, things are always complex. Aaron, there's a question for you. People are obviously very interested in the ultimate application of devices that you're working on. Once your study has been approved as a clinical treatment, the question goes, do you have any idea how affordable such a device might be?
Yeah, it's a tough question. I know I'll answer it in a very summarized manner. It will be a price that insurance companies and hospital systems will pay for patients. So we have to find, there's negotiations happening right now in the background, years in advance, finding the sweet spot for what can be charged. And that's actually, that will go into actually how complex the device goes. Because of course, for a company to run all the trials and take all the risk, there will need to be some profit in it eventually. So it will be in a price range that's affordable to healthcare systems. And that's kind of what we're working with right now, discussing with them.
Good. Given that we're in a roundtable here, and yet the subjects that you three have presented are very, very different, I'd like to pose the same kind of question to each of you in turn. And, considering that the audience here is not just interested in what you've done already, but clearly is interested in where you're going. Like, what is the next sort of crucial step? And it may even be a different approach to a different issue. But Matthew, could I start with you? You've kind of led us up to the brink. You've shown us the iCube that can, respond with some small degree of delay to switches in conversation. Where do you want to go from here?
Yeah, that's a great question. So one of the big problems that we're working on as a field in robotics is how to integrate the fantastic new developments in deep neural networks into robotic systems themselves. So a lot of people kind of think of robotics and AI as the same field, and yet it turns out that robotics engineers are really reluctant to start using deep neural networks because they don't know how they work inside. And they'd much rather have very deterministic engineering equation solutions to their robots rather than just trust the black box of the neural network. So we're at the point now of trying to integrate deep learning into actual autonomous systems. That's going to be a real challenge.
Aaron, where are you heading?
We're at the kind of level of discovery at the final frontier right now, but we're heading into building off of our tech. There's a few different ways. I mentioned the classic clinical trial to get this to market, which is happening soon and will probably start in April, May here in Calgary. We also are working in my basic science lab to understand if we can use some cell type specific approaches to only activate neurons that are responsible for blood pressure control and have no other effect on any other systems within the spinal cord. So that requires some single cell sequencing and developing of new lines, and then probably optogenetic control of those cells eventually. And then the third step is really going to be the work with DARPA, where we're developing what's called the SOS, the system of systems. So it'll be a series of closed loop systems controlling bladder, temperature, spinal cord perfusion, and blood pressure, all within closed loop, and then all within the system of systems. So a little bit space age, but this is what we're trying to bring forward, is almost a fully externalized autonomic nervous system, a fully implantable autonomic nervous system for people that have lost that.
And so addressing not just the blood pressure issue, but other issues that patients that have lost neurological control face.
Exactly. The patient priorities.
Thank you. Anastassia, where are you heading? It's pretty clear, I should say this of all of you, that there are many ways you could head. So Anastassia, what's the next step?
Yeah, so we picked multiple sclerosis first because multiple sclerosis is sometimes called Canada's disease. And so we felt like it was so important for us to start with this and see if we can really use preclinical mouse models of MS first and foremost. But as I've already mentioned, we have already ongoing collaborations for testing our compounds in mouse models of schizophrenia. And I haven't also mentioned, but we have a very specific rare disease mouse model. It's called KBG syndrome. And I know that we have a few patients here in Alberta, but they're all over the world. And we're trying to do the same thing. Can we target sort of myelination with some of the MS drugs or our MS themed compounds to increase myelination and hopefully maybe rescue some of the behavioral deficits in some of these neurodevelopmental disorders? It's a very big, very bold vision. But that's where we're heading. That's where we're tying neurodevelopment and neurodegeneration together. Sometimes it's bewildering to some people while we're doing that, but we have a vision behind it. And so the vision, I think, thinking 10 years from now is really looking at the pharmacological compounds, because even with fractalkine, everyone was looking at inhibiting that pathway. No one has ever looked at activating that pathway, which is what we're doing. And so there is no actually tested pharmacological compounds to activate that receptor. And so we are going to be looking at working with pharma companies to develop these new drugs, or maybe if we're going to rediscover some other ligands, maybe we can repurpose some of the old tested drugs as well. And so that's where we're really seeing ourselves heading into the future.
So Matthew, of the three of you, you're not at the stage where you're actually working with people to try and help people, but you did say at the beginning that one of the goals was to make it easier for humans to interact with robots. So we're already talking 10 years out, as Anastassia just said. So how can you sort of project for us where you think the kind of work you're doing to facilitate these interactions could lead to?
Yeah, sure. It's a great question. So one of the things that I find surprising is that there's tens of millions of robots in the world now. And there's been dramatic advances in machine vision. So mainly in the realm of self-driving cars.
Right.
But it turns out that almost no robots in the world can hear. And the inability to hear for a human is a substantial disability called deafness. And yet most robots don't hear at all. And so I think the challenge that we're really interested in is to sort of deploy auditory artificial intelligence into robotics so that you should be able to just talk to your robot. That's what humans use speech for. We exchange information really fast using speech, but you can't talk to a robot. So we're working on that, like implementing auditory AI so that you should be able to talk to your robot.
I'd like to just, in the last couple of minutes of our session, just pick up on that 10-year now. Do you actually think about what might be happening 10 years from now, or do you say that because you know there's so much more work to do that it'll probably take 10 years? I know that's kind of an unusual question, but if someone would take a shot at it, are you actually looking out that far?
I definitely am looking 10 years ahead for sure. Yeah.
And what are you seeing there? See, you shouldn't have answered.
Yeah. Well, I mean, I think the merger of AI and robotics is something that we need to move slowly towards, carefully towards. And I think there'll be truly usable robots in the future that are going to be interactive. I don't think that they're necessarily going to all be of the humanoid type. I think what you're going to find is robots more like Roombas that take care of specific tasks in your environment. One place that we have to be very, very careful of is in the development of robotic systems in weapons systems. And so a lot of us in the field of robotics have sort of disavowed any interest in weaponizing a robot. And so that's something that I think the whole world needs to be watching as a technology in the next 10 years to keep that from ever happening. There's no role for AI in weaponization.
But you have seen the Boston Dynamics videos, right?
Oh, sure. Yeah. And interestingly, those robots were developed initially by DARPA, Aaron, just so you know.
Yes,
10 years out, Aaron.
I mean, I can speak to my technology and where we think we'll be at. I know that we have tight milestones that we're delivering on for the U.S. military, and I don't see a sense that we'll break those. So I know within 5 to 10 years, or I feel certain that in 5 to 10 years, we will have this technology in the hands and in the bodies of folks with spinal cord injury. And it's not just me saying that. I mean, many of the frontline physicians treating these patients believe this will be the standard of care in that timeline. So I feel strongly about that. Philosophically, about where the world will be in 10 years in terms of technology, I mean, I tend to agree with Matt. And the other thing I'll add is that I think there'll be a lot of misused point and click artificial intelligence and neural network tools that people won't necessarily have really understanding of how it's working, but there'll be a phase where we will make conclusions based on those things and those computational models without truly understanding the models and how much error and nuance can go into those models. I don't know what you think about that, Matt, but the release of kind of point and click neural networks that's the voice of God in some people's minds is challenging, and I think it's going to be a milestone we have socially.
Yeah, I think it's an important thing to keep in mind that neural networks are fabulously good at doing what we've trained them to do, and that's it. So we have to be aware of what we're training them to do and what they can't do.
Just a few, very quickly, Anastassia, 10 years?
If I can sort of pinpoint that and sort of do an outward, to the public and to everyone in terms of the regenerative medicine research in general in 10 years, I think that one of the messages that I keep pounding over and over again, it's important to invest into fundamental and translational research. And if we don't discover anything fundamental, what are we going to translate, right? And so I hope that in 10 years, we are going to have good support for both streams, so we actually can work with the clinics and we can work with basic scientists. So it's kind of like a global approach, but I feel like it's such an important message to deliver, which probably is spanning not just regenerative medicine, it's probably going to all areas of science. So
good. Well, thank you all very much, Dr. Voronova, Dr. Phillips, and Dr. Tata. That brings this session to an end. We're taking a 20-minute break. For some of you in some time zones, that's not a lunch break, but for us here in Alberta, it is. And we'll see you back here at 12:45 Mountain Standard Time. Thanks very much, and thank all of you again very much.
thank you very much.
Thank you.
Thanks.